Hello, thank you for joining us on our webinar today. Today we're going to talk about imaging of nanoparticles and validation of their specificity in preclinical small animal models. My name is Seth Gannon, and I'm the In-Vivo product manager here at Terrestrial Molecular Imaging. Very briefly today, we're going to talk about in-vivo imaging and then the assessment of nanoparticle delivery, including validation of nanoparticles, their targeting specificity, and how they work in vivo. A very brief introduction to imaging sciences. All imaging sciences, really all imaging sciences, are unified in the fact that they detect light at some wavelength, and then we're trying to understand where that signal is coming from and what it means. As you move from microscopy to small animal imaging to astronomy, you sometimes go over vast ranges of scales, as well as different artifacts that you have to correct for, and different controls in your experiment to control for those potential artifacts. Today we're going to talk about small animal molecular imaging, although you'll see a few illustrations of how small animal molecular imaging and fluorescence microscopy in particular can interact quite well to get a complete picture of your experiment. When I conduct an imaging experiment, one of the most important reasons is that these are non-invasive studies, so you get a true history of the disease, the developmental process, or in-vivo distribution of the probe that you're tracking. You don't have to sacrifice your animals at each time point that you're interested in studying. This allows you to improve statistical power because of the fact you can switch to statistical tests such as test with replacement, which have improved power over standard cross-sectional studies. Not to mention the fact that you really just have a better data set when you have a longitudinal study than a cross-sectional study. It also allows you to study molecules and molecular events both in time and in space. So what do I mean by that? As you're observing an animal in your small animal imager like the In-Vivo MSFX Pro, you can see where the lights are turning on. Is it turning on in the leg, in the heart, in the chest, or tumor? And you can see how that moves around in space as time changes. At time zero, your probe may in fact be distributed throughout the body, but by 20 minutes or a half hour later, it's now localized itself to the tumor site or to a site of inflammation, for example. Ultimately, we want to utilize imaging to be able to quantify as best we can biological processes such as biochemical pathways at the cellular and tissue level in the most relevant context, that is, all the complexities of a living animal, including periprinotic and signaling. So then people are familiar with classic molecular biology techniques such as multiplex fluorescent vector blocks, or immunofluorescence assays in cells, or well-plate assays. However, some people may be new to the field of molecular imaging. Here, you're looking at a fluorescent reporter in the context of a whole animal reporting on some molecular event, in this case, the presence of anionic phospholipids in the context of a live animal. We can also utilize this with activatable probes as well as with radio tracers to look at things like metabolism, for example. You can also, of course, study phenotypes using anatomical imaging. Here's an example of a human imaging study to look at anatomy, but you can also conduct many types of anatomical studies and imaging studies in small animals through use of a high-resolution X-ray system. So essentially, we see two very important requirements for in-vivo imaging. The first is to observe the bulk physiology and record the physiology and anatomy as best you can with an image, whether it's a reflectance image or a high-resolution X-ray image. You can then zoom in on these structures through our optical zoom and really in more detail both observe and record and then quantify changes in phenotype using the X-ray. Next, you can then detect and co-register through molecular signals through the use of fluorescent, luminescent, or radioisotopic probes to these parts of anatomy under study. You could also then correlate them to changes in bone or soft tissue phenotype through quantification of the X-ray in the frame of previous images. So more specifically for this presentation, what are people interested in utilizing nanoparticles in vivo? There are a few main reasons. The first is to improve delivery of conventional drugs or of larger non-classical therapeutics such as siRNAs or proteins or protein fragments. One could also utilize nanoparticles complexed with multiple radiotherapeutics in order to effectively kill a tissue, most likely, in that case, some sort of neoplastic tissue. Others are interested in developing nanoparticles specifically to act as imaging agents in order to more readily assess the presence or absence of biomarkers or molecular events in vivo. These could be targeted probes where you coat the surface of your nanoparticle with a ligand that's targeted to a specific receptor, or it could be smarter algorithmic probes where somehow the nanoparticle, when it's acted upon by an enzyme, changes its light output. This is particularly relevant for fluorescent and chemiluminescent probes. The last way you can utilize, one of the main reasons that people might want to utilize nanoparticles is because it's relatively large compared with other small molecules. Thus you can readily think about combining both targeting agents, imaging agents, and drugs all in the context of a single nanoparticle. That may effectively allows you to both to image where that drug is being targeted in the body at the same time you're delivering it. This has an application to a wide variety of different therapeutic areas, including oncology, inflammatory diseases like arthritis, or ischemic injury. Right now, nanoparticles are primarily being utilized by preclinical researchers, although there is ongoing translational research with nanoparticles. Really, there hasn't been a lot of these nanoparticles reach the clinical imaging or clinical therapeutic realm yet, although things are moving in that direction. That brings us to the main real thesis of this talk, and that is, why are people interested in studying nanoparticles in vivo at all? And how can studying them using infrared fluorescence in particular be useful to you as a researcher? Infrared fluorescent imaging of nanoparticles allows you to really rapidly validate the delivery and efficacy and specificity of your nanoparticles. It allows you to look at the approximate pharmacokinetic profile of your nanoparticle as you change its surface chemistry or its optimal targeting times depending on changes in surface chemistry. Once you've optimized these parameters in your small animal model using fluorescence, where you can image up to 5 mice at a time, you can then conceivably move on to imaging in other modalities for much more quantitative assessment. For example, labeling them with a radiotracer and imaging them in a PET spec and CT system like the idea available from FirstStream. So first, let's look at the first topic I talked about, which is how can nanoparticles help us with improving the delivery of drugs to target sites, and how can molecular imaging help you validate that delivery? In 2009, this group was interested in creating liposomes that were in the nanometer range of sizes for improving delivery and variety of drugs. The key thing they wanted to know is whether these liposomes, one, reach their target at all, and two, where else were they primarily accumulated in the body, so they can more effectively look for off-target effects later, as they were found from the drugs. The nice thing about liposomes is they're quite readily labeled with a dye called DIR, which is available from Life Technology, Bruker. In this case, you have a near-infrared fluorophore that has a little tail that readily inserts into these liposomes, and the fluorophore sits on the outside surface. That allows it to become very bright and fluorescent in the near IR, and allows you to label your nanoparticles that have lipids on the surface quite readily without having to have a bioconjugate chemist at your site. It's really quite easy to shake and bake these together. So they injected their neutrophil-labeled probes IV, and they wanted to see where they went. This group ran a very nice control, a very important one, which is to image a mouse that is naive or hasn't been injected with the infrared, within fluorophore group. And that allows you to understand the autofluorescence level curve in this mouse. In the EIR, you minimize your autofluorescence, but there is a little bit of it there, and it's important to understand where that is. So we then injected the liposome IV, and we were able to find that these one were primarily taken up by the liver, but two were actually taken up at an acidic site. One can quite readily tell the uptake in the liver by finding the ones in the animal and looking, which is easy by X-ray, and looking just below the last ribs. So you can co-localize this signal with the X-ray to understand this is not very likely going to the liver. You can now readily localize this signal to the knee joint of the mouse, again because you have the X-ray there in order to understand where that signal is coming from. And then one likes to confirm, well, okay, I know that my nanoparticles have the opportunity to one, get to the liver, and two, get to the relevant tissues, including the athletic site. Is there improved therapeutic delivery of these drugs? So indeed, when you compare the liposome complex drugs to those that are not complex in the liposome, you see a significant improvement in the severity of disease in these mice. Another important variable of drug delivery is putting drugs across the membrane in cells that overexpress multi-drug resistance pumps. One of the most pervasive is MDR1-PGP. This pump is particularly notorious because it's often overexpressed in cancer cells and will prevent the incorporation of cationic phospholipid cationic drugs that are soluble in lipids. The good examples of these are paclitaxel and ceramide. However, nanoparticles, if they're properly formulated, may be able to circumvent MDR1 by entering the cell through other mechanisms such as microphenophotosis or endocytosis, and then delivering those cells into the cytosol, making the drugs much more toxic again. So first they studied whether the paclitaxel or ceramide-labeled nanoparticles were able to more effectively kill their cells. And in fact, you can see this is the case. However, we then have another very important question. We know that these small molecules can get to the tumors in vivo, but now when we loaded up a nanoparticle with these probes, are they still able to get to the tumor or not? And that's a very important question because there really are two forces that are continuously at work in tumors. One is what's called enhanced permeability and retention, which improves the abilities of nanoparticles to be delivered to cells. Fighting against this effect, which is an effect that's been known for years, which is a significant increase in temperature pressure that's pushing out on these large nanoparticles. So it's always a balance, and important to check to see which is winning in your particular phenotype. And this group shows quite nicely that we're getting uptake of the fluorescent-emitting nanoparticles into the tumor. Now, an interesting point about what these guys are doing, very few people show the full dynamic range of their image in their papers. And that's also a possibility, as you can see here. You can show, as the previous group did, the image scaled to the autofluorescence and a control arm, or you can show the full dynamic range. The key is, both groups properly show the scale bars here. First thing we'll move on to from just really showing that you can more effectively deliver nanoparticles without any carbon agents, to looking at nanoparticles that are specifically targeted to receptors on the surface of cells in probes that are activated in the presence of an enzyme. This is a very nice piece of work here from Cancer Research, where they wanted to create nanoparticles that were compatible with many different modalities. We used an iron oxide core that would be compatible with MRI and at much higher concentrations X-ray imaging. We also labeled the surface of these nanoparticles with Psi-5.5, which is very amenable to in-vivo imaging, as well as efficacy, which is much more appropriate for microscopy or fine sorting. I also labeled the surface of these nanoparticles with a peptide that targeted the MUC1 antigen. We then wanted to validate first in vitro and then in vivo that these nanoparticles can be selectively targeted to these antigens. We started off first looking at microscopy, and I'm looking to see if a wide variety of cells, in fact, were labeled with this nanoparticle. You can see that both in the 50 and the 5.5 channel, but many different cells were labeled with these nanoparticles. The 87 control cells, however, were not labeled with them. Looking at these images, it looks like there are areas of internalization of these nanoparticles. However, one would need confocal microscopy to really prove that, in fact, these were on the interior of the cells. What's important for this study is that the MUC1 antigen is very broad-based. So you can see in cell lines such as CAPN2s as well as HT29s. We then wanted to go in vivo using a subcutaneous tumor implant model to determine the specificity of their probe in a small animal. So here on the right they have a MUC1 positive antigen cell line, and on the left a MUC1 minus, which is what would be known as a genetic control, where your target of interest is expressed in one cell line and in one place, but not in the other. This particular experiment is quite eloquently designed because within the same animal, you can look at the target to non-target ratio or specificity quite readily. Both of these tumors are seeing the same injection at the same time because they're within the same animal. So as you can really see from this image, you get significant uptake of the nanoparticle in the MUC1 positive cells, but not in the MUC1 minus. An important part of any in vivo experiment is ex vivo analysis after the fact. You can see here that when we extracted the tumors and imaged them after they've been cut on the surface of the imagery, that there's much higher uptake of the nanoparticles in the MUC1 positive cells than in the MUC1 negatives, and definitely more than in a controlled muscle. We also then went on to show that these cells were labeled by fluorescence microscopy, that they weren't just extracellular accumulation of the probe, for example, in the vasculature of the tumor. So a very powerful point about fluorescence versus luminescence. With fluorescence, you can quite readily cycle back and forth between fluorescence microscopy, in vivo imaging, and then back to fluorescence microscopy as facts. Why is that important? Well, it allows you to image things, one, as you see from in vivo, on the whole animal or sub-organ scale, and then without having to change probes, or without needing to resort to extra staining protocols, you can directly image these at the cellular and subcellular level using fluorescence microscopy. So then wanted to ask the question, okay, we're able to prove quite rapidly and easily that this nanoparticle was specifically targeted using fluorescence imaging. Will this blow up in a 3D tomography experiment, in this case using small animal MRI? In small animal MRI, it's the opposite of a fluorescence experiment for this particular reporter. When the nanoparticle is taken out of a tumor, it actually causes a dark spot to appear. So they took a pre-contrast and a post-contrast image, and you can quite readily see that, particularly in this view, that in the MUC1 positive cells, you got significant darkening of the tumor than in the MUC1 minus cells. This is a nice example of showing how when you have a multimodal nanoparticle, you can go not only between scales, as we saw between microscopy and whole animal imaging, but also between modalities such as fluorescence and MRI, all using exactly the same probe. Because each instrument has its own pros and cons and information that you learn from each experiment. But you never have to wonder, well, has my data changed because my probe has changed? That's not the case because we have the same probe in each experiment. Next, I'd like to move on to a short vignette illustrating all of the different really critical controls that one should think about doing when you're conducting an in vivo validation of nanoparticle study. This particular group is in fact looking at targeting peptides and then labeling them with stratification streptavidin, but all of the principles are the same. As you can see in panel E, they have a very nice targeted uptake of their construct. So you see a bright spot at the tumor site, but not really anywhere else. However, the question then is, how specific is my nanoparticle? They answer that question by conducting a single experiment, such as one showing the autofluorescence of the animal and tumor is significantly lower than what they observed. That's a simple HT experiment. Next, we move on to looking at the dye alone. And that dye is also significantly lower, less taken up than the targeted construct. Tumors actually behave very strangely because they're not all tissue, and take up a large variety of large particles and dyes quite non-specifically. It's important to control for this effect using a dye alone injection control. To show that it was targeted with a specific receptor, they utilized a scrambled peptide that had the fluorophore. In this case, the scrambled peptide had significantly less uptake than the targeted construct. So we know again that this is sequence specific, implying it's receptor specific. We speak then on to conduct another experiment by injecting an antibody that blocks the known binding site of this targeted peptide. We also saw a significant decrease of uptake at the tumor site in the blocked experiment. So we see a really nice set of controls, but actually each one controls for something very different. But you can choose from sort of like a menu when thinking about what is the appropriate control for validating the specificity of nanoparticle in vivo. Very recently published in PNAS by Young Chen and Steve Peralt is a strategy known as multi-stage tumor targeting. In this case, the nanoparticle is not directly conjugated to a reporter or a fluorophore. Instead, it has a linker group, in this case, biotin, which can then be targeted by a second injection of fluorophore-loaded, in this case, streptavidin. The idea is to overcome what we've seen several times already, which is that nanoparticles typically show peak contrast or target to non-target ratios at 24 hours, which may be perfectly acceptable for preclinical applications, but if they were ever needed to be translated to a clinical setting, this time frame is simply too long. A clinician needs to be able to inject and then read out within hours at the most the imaging agent, whether it's a nanoparticle or a small molecule, in their patient. So the idea here is that perhaps the mouse or the patient could be injected 24 hours previously with an unlabeled probe, and then go into the clinical setting and be injected with a second, much more rapidly taken up and clearing imaging agent. The first experiment that we did to validate that they were doing in vivo assembly was to look at those nanoparticles alone labeled with the extra 750. And these have a signal-to-noise ratio of 0.5, which really means they're just fundamentally not detectable. However, whenever they utilize the in vivo assembly strategy, they were able to ignore signal-to-noise ratio. The other important thing is that they were able to quite readily get targeting very rapidly. And you can see here from panel B, when you have in vivo assembly, you can see plateau signal levels at by 50 minutes. And now looking back at this previous slide, if you consider the tumor-to-body ratio of tumor minus body signal, as a measure of specificity, you see a plot of uptake at perhaps 2 hours. Now, again, that would be in the time range that would be much more relevant for a clinically translatable agent. The circulation time of nanoparticles can be overcome another way, at least for imaging agents. And that is to create nanoparticles that are, quote, smart or activatable. In this case, the nanoparticle has 4 forms balanced whenever in close proximity to each other, but whenever a protease comes by and puts a peptide bond, the 4 forms can diffuse away from each other and then become fluorescent. So it doesn't matter how long they circulate, so long as you're seeing it activate, it will be dark everywhere, quote, everywhere, else in the body that the protease isn't present, and then ideally become activated only and bright only in the presence of the protease. So here's an example of a much more recent experiment using a strategy more identical to the previous one with a commercially available probe, MMP-Sense, which is a trademark of Design Medical. And this probe is at broad MMP activation. And this group, we were interested in looking at whether there was MMPs present on the surface of porcine tumors. They were implanted subcutaneously into a mouse and then injected 24 hours prior with the activatable nanoparticle. You can see nice activation at the tumor site and also significant accumulation of fluorescence in the bladder as those free dyes are cleared. The next group of interesting nanoparticles, or ways to think about nanoparticles, are theragnostics, which is a real term for an old concept which is combine a therapeutic agent with an imaging agent. So you can track, use the same nanoparticle, or as I'll show later, the same cell, as a vehicle to diagnose whether there's a problem, because it's specifically targeted somewhere, and then at the same time already begin to deliver the therapy to the affected area. So this group is interested in determining how to use to optimize the surface chemistry of calcium phosphate nanoparticles in order to more effectively target tumors. Here they loaded these calcium phosphate nanoparticles with ICG, 1 as an imaging agent, but 2 as a way to knock load cells with a small molecule that should be drug-like. ICG is certainly not a drug, but it is a small molecule. So 1 injected through ICG to show what that looked like in its distribution and set the flare of the image to that. Then change the surface chemistry in a variety of ways to look at the optimal tumor to non-target ratios, identifying the most specific way to deliver these loaded nanoparticles to a tumor. In this case, you can see the human whole transferred and aberrant calcium phosphate nanoparticles work quite well, and perhaps slightly better than peg-loaded calcium phosphate, peg surface-coated calcium phosphate nanoparticles. We then want to ask a more complex question, which is, okay, the blood-brain barrier is really, it's great because it keeps our brain free from all the many different, many of the small molecule toxins that we encounter on a daily basis. Unfortunately, it also prevents drugs that we need to take from crossing into the brain and many times reaching their target. This will be particularly true with nanoparticles. So they're going to change the surface chemistry of their calcium phosphate nanoparticle again to see if they could find a way to cross the blood-brain barrier. And from a really simple experiment, they were able to image and quite nicely show that these gas-to-antenna peg particles were at least able to selectively be retained in the brain. However, following the effect of fluorescence microscopy would indeed confirm that these particles had gotten across the endothelial cells and, in fact, into the brain. And that would be what's the nice thing about fluorescence again. That experiment, that direct experiment, is a very real possibility. It would be very difficult using other methodologies. One can also utilize the cells themselves, actually, as a way to be able to be a diagnostic. In this case, one can imagine taking stem cells, and in fact this is what happened, loading them with a nanoparticle nanoparticle, or if you want to translate it to humans, a nanoparticle loaded with a heterospec isotope. Those stem cells are then purified based on their stemness and injected into the animal or patient. That allows you to do one of two things. One, to find the ischemic site, because stem cells are known to track to that site, in this case with ischemia, but the site of injury, validate that in fact those stem cells are gotten there, so that if there's no hit there, you may want to re-inject that animal or that patient with the stem cells. And then three, hopefully see those stem cells actually begin to repair the site of injury. And indeed, that's just what happens. Whenever you group sorted for cells that took up the most their infrared nanoparticles, but by 24 hours, they were able to visualize the site of ischemic injury quite nicely, and by 7 days, they could see it kind of really just plain as day. The next question was, were they actually able to repair the injury? Now, left and right's been switched here from the previous slide because this was done in a different type of instrument that's looking for blood flow. And what you can see by day 0, this leg really has no flow, which is denoted by red in a normal leg. That's blue. However, in the animals that have been injected with stem cells that have been enhanced for their stemness, they were able to quite nicely by day 7 completely have their flow restored, which corresponds nicely to what we saw in the imaging experiment, which is that the cells were trafficking there for 24 hours, and by 7 days really had filled up the site. So in conclusion, of all this data, I'd like you to take home a few things. One, labeling nanoparticles with near-infrared fluorophores is readily achievable in a variety of fashions, whether using ICG, 55.5, Alexa 760, there's the DIR, there's a myriad of ways that one can label nanoparticles and track them quite easily in vivo. This allows you to rapidly assess what's the optimum imaging time for your nanoparticle, and is your nanoparticle really specific through the use of the appropriate controls. What's great about small animal imaging is you can image up to 5 mice at a time in an instrument like the NSFX Pro. So you can validate, have all the controls all in the same image to really nail down just how specific and at what time do you see maximum specificity with your nanoparticles. Multimodal nanoparticles, such as those that contain fluorophores or different colors of fluorophores, as well as contrast agents for MRI or PET spectra CT, can be very intriguing because, particularly in the preclinical setting, it allows you to directly compare data between different scales, for example, going back and forth to micronic to whole animal imaging, as well as different imaging modalities, ones that may be 3D versus 2D, and you never have to worry about whether the data is changing because you're switching your probe, or your specificity has changed because you're switching your probe. You don't have to worry about that because it's always the same nanoparticle. And finally, I feel like really the full promise of nanoparticles for in-vivo imaging and therapeutics has yet to be achieved. A lot of this work has been done on delivering nanoparticles to tumors where you have competing forces, including your target molarity, enhanced permeability and retention, and increased turgor pressure in the center of tumors. Nanoparticles may be more effective at areas that are much better perfused and don't have that increase in turgor pressure. So I think there's a lot of opportunity for you in in-vivo imaging to rapidly validate when nanoparticles can be effective and when they're going to be specific. So I'd like to thank you all very much for your attention, and I'd be glad to take any questions that you might have.